On to our lecture. Alec here is giving a lecture on species loss, community collapse, and ecosystem recovery during times of mass extinction, which, if you've read all the blurb, is quite interesting. Alec is an associate professor of paleobiology at the University of Leeds and leads the Deep Bio at Leeds Research Group. Got that? The group's research focuses on large-scale macroevolutionary and macroecological processes through deep time and the accuracy of biodiversity data obtained from the fossil record. That's quite interesting, you're getting biodiversity from the fossil record. Yeah. Alec is a graduate of the University of Sheffield, and he has a PhD from the University of Bristol. Prior to his faculty appointment, he was a Leverhulme Early Career Fellow at Leeds and was previously a Royal Commission for the Exhibition of 1851 Research Fellow at the Milner Centre for Evolution at the University of Bath. So over to Alec to tell us all about evolution and extinction.
Yes, thank you very much. Hey up, everyone. Thanks for coming. So I'm here today to give this talk, and it's part of the Paleontological Association Innovation in Paleontology lecture series. And my official title for giving this lecture is the Paleontological Association Exceptional Lecturer for 24-25. So you can give a bit of feedback to Pallas at the end to say if I am actually exceptional or not. If you ask my students back at Leeds, they'll probably say it's a bit more like just above average, I would imagine. But we'll see. We'll see how I do tonight, eh? And just before I move on, I'd just like to give credit to this fantastic artwork that I've used on my slides, because I don't have a lot of fantastic artwork in my research. And there's a lot of wiggly lines, which I'll try and make as accessible to you guys as possible. But this is James McKay. He's a paleo artist. He painted this fantastic reconstruction of the ocean floor during the early Jurassic of Yorkshire. And he has a book that you can buy on the Paleontological Association website with all this fantastic art in. So if you like the look of that, go and check it out. Okay, so a bit about me. So thanks for the introduction. I think this has already all been said. So I'm based at the University of Leeds. My research focuses on sort of macroevolution, so these large-scale evolutionary patterns in the fossil record, but also from an ecological perspective. So I'm actually an ecologist by training. So I'm interested in how these extinct organisms lived, how they interacted with each other. I also do a bit of biogeography, so I look at spatial patterns as well as patterns through time. But most of my research is done through the case studies of mass extinction events. So these massive losses of biodiversity deep in Earth history that we get preserved in the fossil record. So I'm part of the Paleo at Leeds group. There's about 8 or 9 of us, permanent academic staff with associated research groups. And my own research group is the Deep Bio at Leeds group, where we, as a group, our focus is modeling ecological and evolutionary processes through Earth history. 3 of my PhD students, Amy, Lydia, and Annabelle, I'll be talking a lot about their work today. So just to give them the credit that this isn't all work that I've done by myself. It's a collaborative effort, and a lot of it led by these fantastic early career researchers in my group at Leeds. It's also a massive community effort, the sort of work that I do. It's sort of big picture science, and it involves lots of different people collaborating from different disciplines. So among the pictures here, we have paleontologists, we have geologists, we have marine biologists, we have mathematicians. That's about it. But these people, I won't name them all now, but they'll crop up throughout the talk, and I'll give them a name check then. Also, a little bit more about me, because I always like to learn a bit more about people as well as the work they do. So that's my job, and everything I'll talk about in the rest of the talk is going to be about the stuff I do at work. But I have a life outside work, and that's mostly taken up with my family. So my wife Jen and my 2 boys, Henry and James. And I grew up in a mining town in South Yorkshire. Football's in my blood. So when I'm not working or spending time with my family, I'm still spending time with the older kid, because I'm also a junior football coach and referee. So when I'm not thinking about mass extinctions, I'm usually desperately thinking about football formations for 9-a-side football and working out how we can avoid losing 12-3 again, like we did last weekend. So what's in the talk today? I'm going to introduce mass extinctions and tell you a little bit about what we know about them. And then I'll say what we don't know about mass extinctions. I'll talk a bit about what my group do to try and answer some of those things that we don't know, which is modeling community structure in the fossil record. I'll run through a few case studies of the sort of work that we've been getting up to and give you a little bit of a sneak preview very briefly at the end of what we're planning to do over the next couple of years or so. So first of all, what are mass extinctions? So mass extinctions are a sharp increase in the rate of extinction over a short period of geological time. Now, you guys all know a lot about geology, right? So I won't have to explain like I do to the biologists what a short period of geological time is, right? So I'll just gloss over that. And the key thing about this increased extinction rate is that it results in a significant and prolonged reduction in global biodiversity. So we're not only losing things, it's actually having an impact on the number of species that are actually on our planet. And as paleobiologists, we define that by extinction rates. So the number of taxa going extinct per unit of time, usually per million years. And we can work this out from looking at fossils and working out which ones disappear at certain intervals through Earth history, and then we can plot that through time. And we can compare it to something called background extinction. So extinction is a normal evolutionary process. Things evolve, they diversify, and eventually they go extinct. No species tends to last forever. So background extinction is the amount of extinction we would expect to see under normal evolutionary processes for any given unit of geological time. It seems like it declines through time. We don't really know why. I'm not going to talk about that, but if you want to ask me later, I can come out with some wild hypotheses. And when our red wiggly line of extinction rate sticks out above background extinction, that's when more things are going extinct over a short period of geological time than we would expect to do under normal sort of macroevolutionary processes. And those are our mass extinction events. So I usually do what most sensible quantitative paleobiologists do, and that's remove the Cambrian because it's weird and you get very high extinction rates and very high origination rates, and it's just all a bit chaotic. So we'll just ignore that. Sorry if that upsets anyone. And then what we're left with are these peaks of increased extinction rate. You might have heard of the big five mass extinctions. So the late Ordovician, the late Devonian, the late Permian, the late Triassic, and the Cretaceous Paleogene. But you'll notice there's other intervals as well, where there's more things going extinct than we would expect to go extinct. And that haven't actually just been five mass extinctions, there's been quite a few. And extinction kind of works on a bit of a continuum from very low background rates all the way up to the major mass extinctions. And there's sort of a few in between events as well. We can also measure mass extinctions by extinction magnitude. So rates, quite difficult to sort of conceptualize in our heads in terms of how bad these events were, because you'll get an extinction rate of something like 3.61. Doesn't really mean much if you as an actual number. But we can also take our extinctions as a point in time and look at how many things as a proportion of all animals, for example, go extinct across a boundary. And that gives you extinction magnitude. So in the marine realm, so our marine invertebrate fossil records, these are the numbers of the percentage of genera that go extinct over these mass extinction events. It ranges on the big five from 40% of the KPG all the way up to over 80% for the late Permian mass extinction. So these are quite severe losses of global biodiversity. And these are those figures that are traditionally used to define our mass extinction events. So what do we know about mass extinctions? So from real in-depth study of the fossil record, we've got a really good handle now on when they happened, and we know there were more than five, and we know a lot more about some of these minor extinction events than we used to as well. We've got a pretty reasonable handle on how bad they were in terms of extinction rates and in terms of extinction magnitude. Through loads of research, through Earth system science, geochemical records, sedimentological records, we've got a pretty good handle now on what caused them. So most of our mass extinction events are associated with rapid environmental change, born out of Earth system changes. The ones I study are mostly hyperthermal warming events associated with massive volcanism, continental scale volcanism, which causes rapid greenhouse warming and a whole host of other environmental cascades. And from looking directly at the fossil record, we can work out what things went extinct. So we can work out which groups of organisms disappear at different extinction events. With a little bit of ecological investigation, we can also start to piece together why these things went extinct. So we can look at the organisms that go extinct, we can look at their ecology, their ecological traits, how they lived, and then we can start to say something about which modes of life, which ecological niches were selected against. So we know quite a lot. Another question is, why do we care? This happened like millions and millions and millions of years ago. Is it really that relevant to what's happening today? So despite being really interesting in itself, which I'm sure all you guys will agree with, it's also really important that we understand how our biosphere, how the living world responds to major environmental changes that are happening very, very quickly, because now, because of us, they're also happening really, really quickly. The problem with our ecological records, so records of things that are living today, is that they cannot really inform us, one, about long timescales, because we've only been doing detailed ecological analyses for a century or so, but they also can't inform us extinction scenarios under future warming predictions. So the sort of levels of warming we're expecting over the coming centuries have never been experienced by us as a species, that are known, let alone been recorded by us as a species. But the fossil record has experienced loads of these different events. You can think of them as experiments that have already been run. All we've got to do is piece together that only direct source of evidence of extinction in response to extreme warming from what is somewhat fragmentary fossil record. So that's why it's important. So what don't we know about mass extinction events? There's still quite a lot we don't know, right? And we're focusing on two particular things that I'm then going to talk about relate to the work that we're doing in Leeds. I'll present some of our research that we've been doing over the last few years. So first of all, when we think about evolution, it plays out at the community or population, if we're talking about individual species level. So it's really important to think about when we're thinking about mass extinction events and how they unfold, how we go from functioning biosphere through that collapse to our post-extinction world, devastated post-extinction world. It's actually important not just to think about the environmental abiotic changes that are happening, but also the biotic changes. So what's happening in terms of our community assemblage and our community structure. So what role does community structure play in providing resilience to environmental change? Paleobiologists aren't very good at doing this. We almost always assume that extinctions, when we see them in the fossil record, are primary extinction events. So a primary extinction is when something goes extinct as a direct result of environmental change. So we can demonstrate this with a little bit of a schematic of a hypothetical marine community of 4 organisms. We've got a little clam at the bottom, a scallop, a lobster, and some sort of ray. If we have this community preserved in the fossil record, and over point in time, maybe from one bed to the next, when we're looking at a geological section, we see that, hang on, there we go. The clam and the lobster disappear, they've gone extinct. As paleobiologists, we would always usually assume they're primary extinction events, and then we look to our geochemical, our sedimentological record to look for evidence of what that environmental change was that caused these things to go extinct. However, ecological theory dictates that these things weren't living in just isolation. They were all interacting with each other within a community. So another plausible scenario for that extinction event would be that maybe only the clam goes extinct as a primary extinction, maybe in the response to sort of falling oxygen levels on the seabed. The lobster might be gasping a bit as he's scuttling around, but he survives. But then the problem is his prey source, the clam, has disappeared. So in effect, maybe the lobster went extinct not as a direct environmental effect, but as a secondary extinction, because it can't sustain itself anymore because it's lost all of its prey. We call these secondary extinction cascades, and we don't really understand how these played out during these major mass extinction events in the fossil record. And if we're going to understand how the biosphere responds to these major environmental changes and use it to give us any sort of information about where we might be heading in the future, we need to be able to understand how these biotic interactions shape resilience to extinction. And ecological theory actually suggests that when we're dealing with extinction rates or extinction magnitudes of over 80%, sort of 50 to 80% of taxa, that many of the victims of mass extinction would have perished in these secondary extinction cascades, because you can only cause so many extinctions in a community before it starts to collapse via these secondary cascades. So the other thing we're interested in is once that happens, how do our communities recover in the aftermath of mass extinctions? So from what we know in the fossil record, it looks like mass extinctions result in variable rates of ecosystem recovery, anywhere from less than 1 million to over 20 million years, depending on the event we're looking at or the environment that we're looking at. However, these estimates of recovery are disputed, and we don't really know how to measure it. So an example here from the Permo-Triassic mass extinction, you get different patterns and rates of recovery if you look at different ways of measuring it. So if you just count the number of organisms, generic richness, we see a big drop across the extinction, and then diversity is low through the early Triassic, and then it recovers back to pre-extinction levels probably by the middle Triassic, about 5 to 7 million years after the mass extinction event. But we get quite different patterns if we look at different ways of measuring recovery. So if we look at ecological richness, so functional richness, it's not as big an extinction event in that case. We don't lose as much function as we do species. And actually, the recovery seems like more of a restructuring in that our Triassic recovered communities are actually more functionally rich than our Permian pre-extinction communities. And if we've got other ecological measures like functional evenness, we actually see an increase across the extinction event, which then is maintained throughout that early to middle part of the Triassic. So we don't know how ecosystem structure and function recovers from these extinction events, or whether it returns to pre-extinction states, which is true recovery, or to a new baseline, which might be some element of restructuring. So I've talked about community structure quite a bit. So I just thought I'd explain what we mean by community structure. So communities are complex entities in ecology. They are characterized by structure, which is the composition of the community. So the types of organisms within that community, the quantities of each organism. So that's when we're thinking about things like evenness. It's not just what's there and the diversity of that system, it's the evenness amongst those different organisms within it, but crucially also how they interact with each other. And the thing that my group work on is trophic structure. So this is the tiered organization of organisms within an ecosystem arranged via predator-prey interactions. So what eats what? And these are food webs. So food webs simply are a number of food chains. So we all know like the classic food chains of like worm, bird, eagle or whatever. So once you mesh all those different food chains within a community together, what you get is a highly complex network, like these things here. A lot of complexity. But the good thing is we have lots of tools as ecologists to be able to summarize and analyze these complex entities with some really well-defined key metrics that are actually shown to be reflective of community structure and function. There's one problem. I'm a paleobiologist. I can't just go out into a field and observe what eats what, like you can theoretically in the modern day as a modern community ecologist. So we have an extra question before we even start to think about analyzing community structure in the fossil record is we don't really know what ate what, because that's not preserved usually in the fossil record. One, we could rely on highly accurate depictions like this. And actually, I think it's worth admitting I'm not the first person to think about this. There have been a lot of community trophic structure reconstructions from the fossil record in the published literature. But initially, these were just sort of restricted to visual depictions, which is, I'm not criticizing, I think they're great, depending on expert opinion. So the people working on who knew a lot about the organisms within the communities they studied, based on their expert knowledge, can build like a picture of what ate what. And I think I'm not criticizing these, I think they're probably very accurate, but there's not a lot that we can do in a quantitative sense here. We can't really say anything ecologically about how structure compares to present day or how it's changed through time if we're just basing it on visual depictions. There have been some quantitative analyses and models more recently, but one of the problems with these approaches, even though they're starting to quantify structure and function in the fossil record, is that they still rely on this expert opinion, subjective expert opinion of what eats what within these communities. So what we wanted to do was build a modeling framework where we can take some organisms from the fossil record and base a model whether they'd be likely to eat one another And from that, we can start building up these interaction networks. So I've been working with these two chaps. This is Professor Andrew Beckerman, who's an evolutionary ecologist from the University of Sheffield in the hat there. And this is Dr. Jack Shaw. So Jack, at the time when we were doing this, was a PhD student at Yale University in the US. So there's a lot of food web models out there for modeling modern communities, but they all rely on information that you can't really get from the fossil record. So we wanted some parameters to inform our model that we can get from fossils. So we turned to this diagram, which is the Richard Bamback's ecospace cube, widely used in paleoecology. And basically what you can do here is you can take 3 easily definable traits for marine organisms, combine them together and define a mode of life, so how an organism lived. So the traits we have are tearing. So tearing is where an organism lives, whether it's within the sediment, on the sediment, swimming around in the water. And in terms of trophic ecology, this defines encounter rates quite well. Eat something, you've got to encounter it. You've got to live in the same space as it. Yeah? Feeding, so how an organism actually acquires its food. If you're going to eat something else, you have to be a predator. And we could actually usually define how an organism feeds for most marine invertebrates. Snails are problematic. But for most marine invertebrates, we can define that quite easily from the information preserved in the fossil record. Motility, so whether an organism could move or not, and if so, how quickly. And this is important in trophic ecology because as well as encountering something, if you're living in the same space of it, you've got to be able to catch it. So to be able to catch your prey, you tend to have to be faster than it. And there are obviously some exceptions. But for the most part, if you're faster, you'll be able to catch it, you'll be able to eat it. Now, the Bamback ecospace model is highly simplified. And there's one trait that it misses, which is really, really important in trophic ecology, and that's body size. So we use these 3 traits plus a measure of body size to inform our model. So in the marine realm, body size rules all. And pretty much if you can fit something in your mouth, you'll probably have a go at eating it. And bad things happen to animals that break that rule. So from this really simplistic set of traits, we built the Paleo Food Web Inference model. It runs in the coding environment R, and I'll show you how it works. So all you need to put into the model is a list of organisms for your community. And each of your organisms needs a value for those 4 traits. So it needs a tearing trait, it needs a feeding trait, it needs a motility trait, and it needs a body size trait. Body size can either be numerical in terms of a continuous trait, or it can be categorical. You can say something's big and something's small. And then the model operates as a set of rules. So basically, if you have a certain trait, there are rules as to which organisms with other traits that it can eat. So let's use an example of a tuna and a lobster. In trophic ecology, the arrow denotes the flow of energy. So in this case, we're going to ask, can the lobster eat the tuna? So their traits are tuna's pelagic, lobster's superficial, lives on the seabed, tuna's fast, very fast, tuna's. The lobster's pretty slow, just crawls about. The tuna's very large, big fish. Lobster's pretty big, it's as big as your benthic predators probably get, but it's not as big as the tuna, and they're both predators. So when we put this into the model, it'll run through the different rules that make up the parameters. The lobster can't reach the tuna. The tuna's zipping around in the open ocean, the lobster's on the seafloor. So that rule is violated. It's too slow, can't catch the big fast tuna. So that rule is also not realized, and it's smaller than it. So even if it was living in the same space of it, it could catch it. There's no way it's going to be able to subdue and consume it. So 3 out of 4 not realized, but they're both predators, and we know that predators can eat other predators. Otherwise, we wouldn't have more than 3 trophic levels. So that rule is allowed, but for a feeding interaction, a possible feeding interaction to happen in the model, you need 4 ticks. The lobster also probably disadvantaged in this example because he appears to have been cooked. Yeah, we'll ignore that one. I didn't realize that when I first put the picture in. So the lobster can't eat the tuna. So it does that for every possible pairwise interaction within the community. And the output is a food web that looks like this, a nice visual one, that displays all the possible feeding interactions within the community. But we also get a whole host of these really useful structural metrics and functional motifs. both at the community, so the whole network level, and for each individual organism within that food web. So we can really sort of dig deep and interrogate what's going on within that community. So using this modeling approach, along with a whole host of other tools, which I'll briefly mention a couple of them maybe in this talk, we've got some big questions we want to answer. So we want to know, how do extinction events play out at the community level? So we know how they play out globally and even regionally, but how do they play out within these community frameworks when we allow things to interact with each other? We want to know how community structure recovers after extinction events. We want to know if mass extinctions, particularly the big ones, like the late Permian, and the ones that sort of define those end of the eras, like the Cretaceous Paleogene mass extinction, do they permanently restructure communities? Do our marine communities, are they structured differently in the Paleozoic compared to today? And we're also interested in, well, Amy, my PhD student, is really interested in how communities respond to the loss of apex predators. So the big things at the top of our marine food webs, which are some of the most endangered and vulnerable species in our oceans today. What happens when we lose them? So to answer some of these questions briefly, I'm going to take you on a catastrophic journey through the Phanerozoic. I'm going to make 4 stops along the way. We're going to stop, first of all, at the end of the Paleozoic, and we're going to look at ecosystem recovery from the Permo-Triassic mass extinction. So those early Mesozoic marine environments, how are they functioning? How are they structured? How does it recover back from the biggest mass extinction in the Phanerozoic? Then we'll go further forward into the Mesozoic, into the early Jurassic, to one of our more minor extinction events. It's called the early Toarcian extinction in the early Jurassic, and we're going to look at community collapse across this event. Then we're going to fast forward right to the end of the Mesozoic and the beginning of the Cenozoic and look at community structural changes across the KPG mass extinction. And then almost at the present day, just a mere 2.5 million years, blink of an eye, into the past, we're going to look at the loss of apex predators across the Pliocene megafaunal extinction. So first of all, let's look at the Permo-Triassic. So this is Annabelle. Annabelle started a PhD in my lab group in October. But before then, she was employed as a research assistant on a Natural Environmental Research Council grant, looking at ecosystem recovery from this massive Permo-Triassic mass extinction. So what do we know about the Permo-Triassic mass extinction? It's probably the most intensely studied extinction event. We know a lot about it. It happened 252 million years ago. We lose about 83% of marine life at the generic level. We think that equates to about 95% of species. It's hard to define it at species level in the fossil record sometimes. We know from some other ecological modeling, well, we think we know, that the post-extinction communities, after we lose a lot of species, it becomes unstable. So the way these interactions are now happening is prone to further collapse if you lose any more species. We don't know quite how long or how it happens, but the recovery interval is long. It takes a long time, with estimates from 5 to 50 million years for full recovery. 50 million years is the entirety of the Triassic, by the way. So what don't we know? How long did ecological recovery take, and how did that recovery unfold? How did it play out within our marine environment? And we approached it from looking at some competing hypotheses that were already out there. So first of all, there's Chen and Benton's model of ecosystem recovery through the early to middle Triassic. And they state that in the immediate aftermath of the mass extinction, which is the bottom slice of this weird sort of cheese pyramid going on, so this is a trophic pyramid, the immediate aftermath of the extinction at the bottom, we've only got primary producers and primary consumers left. So everything's gone extinct, and we've just got a very low diversity community of things sort of photosynthesizing and then things grazing on them and eating. And then it takes time step by time step up till the end pretty much, well, the middle of the middle Triassic, for that whole trophic pyramid to reassemble and the return of large apex predators at the top of our food webs. However, evidence in the fossil record kind of already refutes that idea a little bit, because in 2023, there was a paper published, there was a detailed study of an exceptionally preserved ecosystem from the early Triassic of South China, which showed that actually less than 1 million years after the mass extinction, we've got what appears to be a fully functional trophic pyramid with a big giant coelacanth as your apex predator sitting at the top of it. So even though we're not really testing whether the Chen and Benson idea is true, because we kind of know it isn't, we wanted to see if these communities were actually fully recovered less than 1 million years after the mass extinction, because that'd be quite groundbreaking compared to what we always thought about the long recovery for this event. So what do we want to do? We want to test how marine communities recovered from this extinction event by modeling our food webs through a time series through the Triassic, and then quantify those changes in ecosystem structure and function to look at how that ecological recovery played out. So for the data set, we're working with Haijun Song, who was one of the co-authors on this paper, that suggests recovery was fast, and they've got these snapshots of exceptionally preserved communities that they use, all from South China. And they range from just after the extinction event all the way up into the late Triassic. So Annabelle, with help from Paul Wignall, Triassic mass extinction expert at the University of Leeds, coded up all this data, and then with the help of Andrew, Jack, and myself, bunged it into the PFIM, the Paleo Food Web Inference Model, to do the analysis. And this is what she produced. So here's our time series through across the mass extinction event, which happens at that Permo-Triassic boundary, all the way through the lower, middle, and upper Triassic. Just from looking at that, you don't see that much change, right, visually. So we can confidently say that our less than 1 million years after the mass extinction, we've got a really well-functioning marine community of 4 trophic levels. So we definitely haven't got that devastation just resulting down to just a few primary producers and primary consumers. What we see there's a general trend of increasing alpha diversity within the communities. So alpha diversity is the number of species within a community, which is different to global diversity, which we refer to as ecologists as gamma diversity. Interestingly, though, the alpha diversity trend of increase, which peaks in the Anisian in the middle Triassic, matches what we see globally. So it actually fits the same picture globally as we see within our different communities. So our lower Triassic communities are less diverse than our middle and late Triassic communities. If we look at the structure of these food webs, we see a general trend of increasingly complex food web structure through our time series, but there's a big leap between the lower and middle Triassic. So our first 3 communities from the lower Triassic are a lot less complex than our middle and late Triassic communities. So again, fitting with the idea of about a 5 or 7 million year recovery, but we're starting from a higher functioning baseline than was always thought previously. However, the recovery doesn't tend to peak or finish in the middle Triassic. If we look at certain metrics and motifs that are indicative of vertical complexity, so that's the height, if you would like, of the food web, it continues to increase in complexity into the upper Triassic. So thinking about higher trophic levels as we get more diversity at our higher trophic levels, like our marine reptiles and things like that. And also the number of linear food chains up through the food web continues to increase beyond that middle Triassic. So what our sort of more sophisticated analysis of ecological structure has shown is that actually the recovery is well underway quite quickly after the extinction event, but then there's a big leap after that lower Triassic into the middle Triassic when things get a lot more complex, but then that structural recovery is still going on right up into the late Triassic. An interesting thing about these early Triassic communities, even though we've got 4 trophic levels, what we actually see is a weird top-heavy community where we have quite low diversity at our low trophic levels and higher diversity at our high trophic levels. And that's because in the pelagic realm, where most of our predators are swimming about, we seem to see higher recovery rates than on the seabed, where things are generally lower trophic level. And this is probably to do with sort of prolonged dysoxic conditions through the lower Triassic. And this also backs up what we see in the global record, where we have these weird reversed functional pyramids in our early Triassic oceans. So moving on from recovery, we're moving on chronologically, but now we're actually going to look at an extinction event itself and think about how our communities actually respond to species loss across an extinction event. So we're going to move into the Jurassic, a project that was the first project we worked on using this methodology. So myself and Andrew at Sheffield and my colleague at Leeds, Chris Little, who is an expert on this particular event. So what do we know about the early Toarcian extinction, about 183 million years ago. We lose about 25% of marine genera globally, so it's much less severe global event than the Permo-Triassic that we just looked at. But it's very geographically variable, and that's particularly evident when we look at sections out on the Yorkshire coast, so the Cleveland Basin near Whitby, where it is incredibly intense, this extinction event. We lose about 80% of marine benthic genera locally in the Cleveland Basin, and it's a highly anoxic. So if you look at our sedimentary section on the right, you can see that black bar. These are black shales, which are deposited under disoxic to anoxic conditions. So it's an isolated ocean basin at this time, which suffered sort of prolonged disoxic conditions, which drove this extinction event. And these disoxic conditions continue after the extinction event as well for a long time, and we get a very long recovery interval locally in Yorkshire. So what don't we really know? So we don't really know how extinction dynamics played out within the community, how we get from that pre-extinction happy place to that post-extinction unhappy place. And we don't know how that ecological recovery unfolds. So our study area, I actually did a bit of field work out here with Chris and some of our students. So this is Robin Hood's Bay, it might be familiar to some of you. And that is Ravenscar Cliff, which is the south end of Robin Hood's Bay. And the extinction beds crop out just behind that cliff, quite low down, so you can actually get up to the cliff and you can sample up bed by bed. It's also a good place to eat fish and chips, that part of the world. So it's a really high resolution data set that we have for this. So our objectives were to test primary extinction trait selectivity. So remember, primary extinction are the things that go extinct because of environmental change. So if we can think about what sort of traits would make organisms vulnerable to extinction, particularly in an environment like this, where we know that the main extinction stressor was most likely a lack of oxygen, we can model that then in a computational sense. We can take our community and we can target things in simulated extinctions that we know would be vulnerable to those sort of stressors, and then we can see what happens to the rest of the community via our secondary extinction cascades. But we also want to look at the recovery as well, so what happened in the aftermath. So the data comes from years and years of study, dating right back to Chris's PhD in the 90s, through Jed. This is Jed, this rather dapper looking chap at the bottom there with a hat. So he did a PhD at Leeds, did lots of field seasons out on the Yorkshire coast. And we had a Paleontological Association summer intern, Carolina, in 2017, who helped us do a little bit of extra sampling. And that's my eldest son, Henry, there, bashing rocks with her in the lab. Probably wasn't supposed to be doing that. And then from that data set, we shoved it all into our model to build some food webs. And then what we did some extinction cascade modeling. So what you do essentially is you take your pre-extinction community, once you've built it from the fossil record, then within our simulated extinction environment in our computer, we can pick things to go extinct. And we can do this randomly, or we can do it, like I said before, from targeting particular traits. And we let the extinction model run until it hits the same level of diversity that we see in our post-extinction community in the fossil record. And then we can do sort of like a matching exercise and see which model of selectivity best reproduces what we actually see in the fossil record. So that's what we did. First of all, what happens across the extinction event? So we go from a diverse community to a low diversity community with a high abundance of opportunists in our post-extinction world. We see increased levels of connectivity. So everything in our post-extinction world is a little bit more densely connected to everything else. So we've got fewer things left, so they become a little bit more dependent on each other in terms of predator-prey interactions. And that increases the generality. So we've got more generalists. It makes sense that a generalist is more likely to survive an extinction event because they can eat anything compared to a specialist. But when that happens, when you get a lot of generalist predators that can eat anything, the few remaining prey are preyed upon more readily. And that's what we refer to as vulnerability. So our vulnerability increases as well. We see an increased mean trophic level and vertical structure, which might seem a bit counterintuitive, but that's because the things living in open water are doing a little bit better than the things living on the disoxic seabed, and they tend to be predatory taxa. So we're increasing our mean trophic level across our entire food web. But we see reduced competition because we're losing a lot of species. So the competition within each broad ecological niche is going down. And we see increased omnivory. So omnivory in a food web sense is feeding across different trophic levels. And we see increased omnivory, we think, because the remaining predators are having to eat different things at different trophic levels to find enough food. The sort of fossil record pattern of this extinction is a loss of large-bodied, infaunal, and motile taxa that live on the seabed, and particularly those predators that are moving around on the seabed trying to find food. So with our extinction dynamic modeling, I won't go into this in too much detail, but put it simply, our simulations that focus on tiering, so selecting our primary extinction towards things that live either on the seabed or in the seabed, produces the best match to our post-extinction community. And we were really pleased about this because that's exactly the result we would expect to see, given that a dysoxic environment is going to be more intense on the seabed and in the sediment than it is, say, in surface waters, where there's that bit of gas exchange still happening with the atmosphere. And it's our benthic predators that suffer the most frequent secondary extinction cascades, so things like our crustaceans, echinoderms, and predatory mollusks. So what happens in the recovery? So we've got our 4 broad time intervals: our pre-extinction meta-web, our post-extinction, our early recovery, and our late recovery. Recovery is delayed, but we see that sort of gradual recovery of diversity and structural recovery, and we reach most of our metrics reach pre-extinction levels by this late recovery interval, and that is no surprise. That's when we return to full oxic conditions. So that prolonged dysoxia is sort of dampening our ecological recovery, particularly in terms of things living on or in the seabed. However, something interesting does happen, and that's, even though most of our metrics sort of recover to pre-extinction levels, some of them that are indicative of increased predation pressure, increased vertical structure, which we can think about might represent energy flow up through the system, actually overshoot our pre-extinction levels, and we have to see a greater complexity within our communities in our late recovery interval. And we wonder, we hypothesize, and it's something we're aiming to test over the next few years, whether this is actually a sort of a broader scale macroevolutionary pattern that we're seeing with the evolution of new predatory groups as we get towards the middle Jurassic, which might be indicative of the Mesozoic marine revolution. Those sort of intense, supposed hypothetical escalation of predator-prey interactions that happens from the middle to late Jurassic onwards. We're going to fast forward the rest of the Mesozoic, though, and maybe we'll have an answer to that question in 3 years' time. And we're going to look at community structural changes now across the KPG mass extinction. So this is work by one of my PhD students, Lydia Woods. So Lydia is funded by the Leverhulme Trust on an extinction studies program. And she's a marine biologist by training and really interested in thinking about how community structure has evolved through time in the oceans, and whether the community structure we see in our modern oceans today, whether that has a defined point in time when it was established. And one of the main candidates for that is the KPG mass extinction, because it was the last major mass extinction to hit our planet. So what do we know about this event? We know it happened 66 million years ago. And of the big 5 mass extinctions, it's now thought to be the least severe in the marine realm. Only 40% of marine genera go extinct. Obviously, it's more famous for what happens on land, where we see the demise of the dinosaurs. So what caused it? We know that there was a big meteorite that hit the Earth on the Yucatan Peninsula, so a bolide impact. But there was also large igneous province volcanism, so one of these big continental scale flood basalt eruptions happening, the Deccan Traps, which can be found in India today. And that was happening at or at least close to the KPG boundary. Most research today firmly supports the asteroid impact as the main driver of mass extinction at the Cretaceous Paleogene, even though the terrestrial record is very patchy and we can't really say for sure. In the marine fossil record, we see a real clear, very sudden extinction signature at the boundary, which is indicative of the bolide impact. But there's still a little bit of debate going on as to whether the KPG mass extinction happened because maybe Deccan volcanism was weakening the biosphere prior to it. So did the Deccan volcanism cause structural changes prior to the KPG bolide impact that maybe these communities would become a little bit less resilient and were more prone to collapse during the impact event. And then we're interested in whether the KPG mass extinction caused a permanent shift in marine community structure. And there's been lots of hypotheses put out there that suggest that maybe the KPG is where, because of this mass extinction event, lots of stuff goes extinct. And in the recovery after that is when we see the origins of sort of modern oceans. And even some people like Jennifer Dunn, who I work closely with, went even further. In 2014, she hypothesized that maybe that's why we haven't had a major mass extinction in the last 66 million years, despite lots of climatic upheavals, because more modern Cenozoic communities are just more resilient to these secondary extinction cascades than more ancient ones were. So Lydia wanted to test some of these things. So she wanted to test whether the KPG mass extinction caused a permanent shift in marine community structure. So do our late Cretaceous communities look very different to our recovered sort of Paleogene communities? And she also wanted to look whether community structure changed prior to the KPG boundary in response to Deccan volcanism. So actually, really detailed marine sections across the KPG aren't that common. But one place that we get really good expanded sections across the KPG boundary from marine settings is in Antarctica. So this little map on the middle panel is Seymour Island. So Seymour Island lies just off the Antarctic Peninsula. Obviously not that accessible, but surprisingly for Antarctica, it's ice-free most of the year. So it's actually just a big lump of mud in the Southern Ocean. So we worked with a couple of experts from the British Antarctic Survey, Rowan Whittle and James Witts, formerly PhD student at the University of Leeds, now a lecturer at the University of Bristol. They've both done field centres on Seymour Island, field seasons on Seymour Island, collecting across kilometres of this expanded section across the KPG boundary. And it's a high latitude environment, but back in the Cretaceous, the world was a lot warmer. So it's a lot more diverse than a high latitude community would be today. So Lydia inherited this data set, supplemented it with extra data from the paleobiology database for the big stuff, so the big reptiles in the Cretaceous. And then we fed it through the PFIM. And this is what her time series communities look like. We have a pre-Deccan community. We have a late Maastrichtian community just prior to the KPG boundary. So they're our Cretaceous ones. And then we have a recovery interval community. So after the mass extinction in the Danian, which is the first time period of the Cenozoic. Then we have a gap because there's a bit of an unconformity going on where we have no marine sediment. And then we have an Eocene section, which would represent by that point, many million years after the mass extinction, a fully recovered community. So what we see, really interesting, a result that none of us expected to see, despite a diversity drop of 86%, most of our structural metrics remain fairly stable across the mass extinction event. We see some changes, but our broad scale measures of how the community is structured remain pretty much the same. There's no significant changes despite losing so many species. Oh, there's me try. And we put this down to something called the skeleton crew hypothesis. So we see this in the global data from the fossil record as well when we look at functional diversity. So across mass extinctions, you can lose a lot of species, but often what we see is we see a loss of functional redundancy. So we're losing species from within broad ecological guilds, but within each of those ecological guilds, which is largely composed of organisms pretty much doing the same thing, a few survive. So what that happens is across these mass extinction events, community function is maintained by just a few species, and that's your skeleton crew. So it's been maintained just by a few species. These communities are really vulnerable to further collapse because if a few of them go extinct, the whole thing can come down. But what we think we're seeing here in the aftermath of this major mass extinction event is a skeleton crew of a few organisms maintaining each functional guild during that early recovery interval. We see an increase in generalism, so more generalists. That's a really common pattern that we see across all of our mass extinctions. But this time we see a decrease in mean and max trophic level and fewer of these small scale interaction motifs within our food webs. This is a different type of extinction to our warming, dysoxia-driven extinctions that we talked about for the previous two case studies. Here, we're losing things from the top of the food web rather than lower down. And the key hypothesis for the KPG extinction is meteorite impact causes sort of nuclear winter, darkness, cold, and it basically shuts down primary productivity. If you shut down primary productivity at the base of the food web, you're not getting energy flow up, and that effect is exacerbated the higher up you go through the food web, so you're going to preferentially lose your apex predators at the top. By the Eocene, everything's pretty much recovered, and that Eocene community is structured in an almost identical way to our late Cretaceous community. So there's no evidence that the KPG actually causes a permanent shift in community structure. It seems like our communities were back to exactly what they were like in the late Cretaceous, and the late Cretaceous communities look very modern in terms of their community structure. Deccan volcanism. So volcanism happens in this time period here, where we see a diversity increase, we see a stability of structural metrics, we see an increase in specialization, and we see an increase in mean and max trophic level, and we see more motifs. So arguably, even though these changes are quite subtle from the pre-Deccan to the Deccan interval, if anything, we're seeing an increase in complexity. So there's no evidence of any sort of nasty effects on ecological structure brought about by Deccan volcanism, which is an interesting question in itself. So other times in Earth history, these large igneous province eruptions cause absolute mayhem, but at the KPG, it seems the effects of Deccan volcanism was negligible. So moving on to the final case study. So we're almost at the present day, well into the Neogene. Just 2.5 million years ago, we're going to look at apex predators disappearing from the marine realm across the Pliocene megafaunal extinction. So this is work by Amy Shipley, one of our Natural Environment Research Council PhD students. So what do we know about this event? So apex predators, first of all, think about apex predators. They're really important moderators of community structure. So they have a sort of capping effect on everything below them. They keep mesopredators populations in check. They're really important for sort of nutrient fluxes between communities. And we don't really know what would happen if we lost them from our modern ocean today. But the fossil record has lots of evidence of what happens when you lose a marine apex predator. We just have to piece that evidence together. And perhaps one of the most recent examples was this Pliocene megafaunal extinction event, where we lose about 36% of marine megafauna across the Plio-Pleistocene boundary. And most charismatic of those faunas is the giant megatooth shark, Otodus megalodon, the Meg, one of the biggest apex predators we've ever had on Earth, as far as we know. What don't we know, though? We don't know what happens when you lose an apex predator, what happens to community structure. And we also don't know if anything replaced the Meg at the top of the Pleistocene food web. So are we just removing this hyper giant apex predator completely, or does that influence sort of your next layer of predatory taxa, many of which are still extant today? So objectives: did the loss of an apex predator have long-term effects on food web structure? And also, what happened to the trophic role occupied by the megalodon after its extinction? So Amy has the luxury of having 6 different academic supervisors for a PhD across a whole range of knowledge. So I'm a paleobiologist. Andrew is an evolutionary ecologist. Jack's our coding guy. Catalina is a marine biologist, but a marine paleobiologist. She's one of the world leading authorities on the megalodon shark. Tracy Hayes, now formerly at Leeds, now at Plymouth, is a micropaleontologist, so she really knows what's going on at the base of the food web. And Jennifer Dunn is also a structural food web expert based in the USA. So Amy benefits from all of this knowledge, but I think she sees it more of a curse than a blessing, because she basically gets 6 sets of differing opinions on everything she writes that she has to try and reconcile. But anyway, it's it's an interesting team to work with. So Amy used data from the Paleobiology database, which is a freely available online repository of fossil occurrences. It's been built up over the course of 30 years or so. And she restricted her area of study to the North Atlantic because it has a much better fossil record than the rest of the world for this time interval. So she built North Atlantic meta communities for before and after the mass extinction event, stuck them into the FIM. Amy makes really nice diagrams, much better than all the other ones I've shown today. So here is her pre-extinction Pliocene food web for the North Atlantic, and here's our post-extinction Pleistocene food web. 6 trophic levels. Anyone who knows anything about modern trophic ecology, in the marine, in the terrestrial realm, you tend to get mostly 4 trophic levels. In the marine realm, you tend to get 5 trophic levels. So we've got an extra trophic level. And that's made up in the Pliocene of 2 organisms. We've got the megalodon up there as this sort of hyper carnivore. And some geochemical evidence backs this up as well. So there's been some sort of nitrogen isotope studies which can tell you about trophic level of megalodon teeth, which put it way above the top oceanic predators today. Is joined by the sperm whale. I'm not going to talk too much about the sperm whale. Modern day sperm whale, the trophic level sort of like ranges from 4.5 to 5.5. But in our models, it's a little bit higher. You can ask me about it at the end if you're really interested. Cross the extinction event, we lose a lot from the top trophic levels. It's a megafaunal extinction, big stuff goes extinct, mostly from trophic levels 4, 5, and 6. But again, our broad scale structural metrics are fairly stable. Doesn't really do that much to community structure because we've got enough of everything from each level. We do see a decrease in vertical complexity because we're losing so much from the top of the food web. That's obviously going to influence the number of food chains and the mean trophic level of the entire community. And again, we see that increase in generalism. So it seems to be our specialists that are selected against across this event. What happens to our top predators? So here we just have the trophic levels of our top predatory taxa across the extinction event from the Pliocene to the Pleistocene. Megalodon goes extinct, another big shark. Paratodus also goes extinct. Carcharodon is your great white. Orcinus, your orcas. Cosmopolitodus, that's a mako shark maybe. Some big porpoise and your sperm whale. We see a general dip. All of our trophic levels go down. And this goes hand in hand with those community level metrics, that less vertical complexity. And the hypothesis for this extinction event is that cooling, as we go into the Pleistocene ice age, has caused a sea level drop. And along with that cooling of the ocean, when we get sea level drop, we see a reduction in the productive continental shelf area. And we know from the fossil record that the megalodon shark was living on these productive continental shelves. That's what it was doing all its hunting. So maybe what happened was there just wasn't enough energy flew through these marine systems anymore to sustain this hyper predator at the top of the food web. But a lot of its prey go extinct as well. So a lot of the big baleen whales that were really diverse in the Miocene and Pliocene go extinct at this point. So maybe that was bringing our megalodon in more competition with the trophic level 5 predators for prey, so things like the great white shark. So maybe some of these ideas about megalodon being killed off by great white sharks, not physically by them, but by being drawn into competition with them, maybe that is what actually happened. And that's shown that nothing is replacing the megalodon at the top of our food web as the apex predator. So, I'm almost talking for an hour. That's a bit too much, isn't it? Sorry about that. So, just briefly, what are we going to be doing next? Because we've left some questions unanswered. So one thing we haven't worked out yet is when did modern marine ecosystem structure originate? So this is what Annabelle's going to be looking at for a PhD. So we know that in the late Cretaceous, our marine communities look very modern. So we have to go further back to find that interval. So maybe in between the Permo-Triassic and the KPG, for the entirety of the Mesozoic, maybe we'll be able to detect some signals of these evolutionary arms races, these escalation events where predator-prey interactions were supposed to intensify. So that's what Annabelle's going to be investigating. Why hasn't there been a major mass extinction in the last 66 million years, despite really profound climatic events like the Paleo-Eocene thermal maximum, the other Eocene warming events? They have all the signatures of an environmental event that should cause a mass extinction event, but they don't. So my postdoc, Baran, who started last January. He's investigating that. He's applying some of these modeling frameworks to test the resilience of communities through the Phanerozoic. But one thing we can't currently do with the models that I've presented today is make a perturbation at the base of our food web. The eagle-eyed amongst you might have noticed that all my food webs look like reverse triangles with a little dot at the bottom. That's our primary production. At the moment, they're just structural topological food webs, so we don't infer anything about biomass. But if we want to cause a bottom-up cascade from the base of our food web, we need to be able to incorporate biomass, primary productivity, and population sizes. So our postdoc in Sheffield, Tanya, who is a food web modeler, is currently adapting existing sort of bioenergetic food webs where we can actually simulate primary productivity, adapting those for use on our fossil data sets. And we're really interested, once we can nail down some of these questions, in trying to apply this to working out how a modern marine system might respond to, say, a Permo-Triassic style mass extinction event. And that's the focus of the grant that we currently have with all these people at Leeds and further afield. We really want to be able to push forward using paleontological data, rich set of evidence from the last 500 or so million years in the Phanerozoic to helping us inform us about our planet today. And I'm 2 seconds over an hour, and I'm going to stop there. Thank you very much.